Today we're going to be talking about sodium valproate and the Wnt Beta-Catenin Pathway, along with androgenetic alopecia. How could this anti-seizure, anti-epileptic medication provide insights into combating hair loss in conditions like male pattern and female pattern baldness? The study found that the sodium valproate group had a statistically significant increase in total hair count compared to the placebo group. This suggests that sodium valproate, or valproic acid, could be an effective treatment for increasing hair count in patients with androgenetic alopecia. Hello everyone, and welcome back to yet another video. When it comes to growth stimulants and hair loss, Minoxidil is considered the gold standard for stimulating hair growth from hair follicles and strengthening existing hair. There are even lesser known growth stimulants that work on different aspects of hair loss, such as prostaglandin analogs like latanoprost, bimatoprost, and travoprost, with travoprost being the strongest out of those three. But there's been some chatter, particularly about an anticonvulsant medication that could possibly reverse some of the downstream effects of DHT on hair follicles genetically predisposed to androgenetic alopecia when applied topically. Today we're going to be talking about sodium valproate and the Wnt Beta-Catenin Pathway, along with androgenetic alopecia. How could this anti-seizure, anti-epileptic medication provide insights into combating hair loss in conditions like male pattern and female pattern baldness? And is it even safe? Before we start, we need to understand the Wnt Beta-Catenin Pathway and review some mechanisms of action when it comes to androgenetic alopecia and what DHT is actually doing in that specific hair loss condition. Now, just to ground ourselves, we can refer to the review titled, quote, Androgen Modulation of the Wnt Beta-Catenin Signaling in Androgenetic Alopecia, unquote, by Premanand and Raj Kumari for our understanding. So essentially, it goes like this. You have free testosterone, which is converted into dihydrotestosterone, also known as DHT, locally in the scalp tissue by an enzyme known as 5-alpha reductase. DHT then binds to androgen receptors. Then, a structure known as a DHT androgen receptor complex is created and then enters the nucleus of the dermopapilla cells, and these cells regulate hair growth. The nucleus stores the DNA, which houses the rules and instructions for building certain proteins and changing cellular behaviors. The DHT androgen receptor complex binds to specific response sites on the DNA sequence. This interaction activates certain genes that in turn trigger inhibitory factors, leading to the suppression of hair growth from the hair follicles. All of this happens to various degrees and extents based on the genetics of any given hair follicle, as some hair follicles on the scalp received more amplified rules and signaling that make them more sensitive to DHT. In healthy hair follicles, the Wnt pathway plays a crucial role by facilitating the transition of hair follicles from the resting phase to the growth phase. Here, beta-catenin has an important role in the pathway. Beta-catenin is a protein that acts as a signal molecule, so this means that it helps to transmit messages from outside of a cell to the inside of the cell. Normally, beta-catenin is regulated by the Wnt pathway. In its absence, though, beta-catenin is broken down. So when that Wnt pathway isn't on and activated, there's no beta-catenin. However, when the Wnt signals, like those from the proteins Wnt3A and Wnt10B, or Wnt3-alpha and Wnt10-beta, which interact with frizzled receptors, and these are specialized proteins on the cell surface that receive Wnt signals, and co-receptors like LRP56, which help to amplify the signals and initiate changes inside the cells. When those Wnt proteins are active, beta-catenin is preserved and can accumulate inside of the cell. Now, this allows for it to enter into the nucleus where your DNA is stored, and it can assist in activating certain genes that promote hair growth and follicle development. This role of beta-catenin is essential for counteracting the hair thinning seen in androgenetic alopecia. So essentially what we can draw from here is that when everything's working correctly, certain genes are activated that initiate the proteins Wnt3A and Wnt10B. And when these proteins are floating around, they interact with these things called frizzled receptors. And these receptors are like little doors or, keyholes that these proteins connect to on cellular surfaces. And once these proteins connect to those specific receptors, it amplifies the release of beta-catenin. So it's saying, okay, let's produce beta-catenin, and then beta-catenin itself causes for a host of hair follicle cells to differentiate and proliferate. So for example, your hair follicle stem cells, for instance, beta-catenin can help energize them to differentiate into different types of cells, like the dermopapilla cell or the keratinocytes. And then there are other Wnt proteins that are involved in hair follicle rejuvenation and hair follicle stem cell proliferation and differentiation, but we'll just stick to the ones that I mentioned for the sake of this video. Now, according to a study by Papukashvili et al. Titled, quote, Perspectives of miRNA targeting DKK1 for developing hair regeneration therapy, unquote, there is a potential therapeutic aspect in combating androgenetic alopecia by targeting components that negatively regulate the Wnt beta-catenin pathway, such as the DKOP1, also known as the DKK1 protein. So with the DKK1 protein, it actually interferes with the Wnt beta-catenin pathway's ability to maintain necessary signaling for hair growth by disrupting the connection that normally enhances the beta-catenin protein's ability to stimulate or activate genes involved with hair growth. This disruption occurs when DKK1 binds to the LRP56 co-receptors. Now, I should remind you that these receptors are usually bound to interact with proteins like Wnt3A or Wnt3-alpha and Wnt10B or Wnt10-beta. And those proteins are the positive signals, right, that actually tells the cells to, hey, continue growing hair. Now, because of genetic instructions, the if-then-else statements that are coded into people that have the genetics for androgenetic alopecia, DHT signals for the production and upregulation of the DKK1 protein and the downregulation of proteins that are good for hair growth, such as the Wnt3A and Wnt10B and beta-catenin proteins. So you're actually seeing DKK1 protein competing with the Wnt proteins for stimulating that same receptor, the LRP56 receptor. And whoever gets that receptor first can signal specific messages first, right? So you have two different sets of information for people that have those genes for androgenetic alopecia. If there's more DKK1 protein hitting that LRP56 receptor first, it's going to signal inhibitory or suppression effects on the hair follicle cells, telling it, hey, stop growing, stop proliferating as frequently, start to suppress your growth. Whereas the other factors, the other Wnt proteins, had they been in abundance, had they been upregulated or not disrupted due to DHT's downstream effects. And by the way, DHT is actually responsible for initiating the instructions in your DNA of these hair follicle cells that causes for the downregulation of those positive Wnt proteins. But yeah, had they been upregulated, those Wnt proteins, then androgenetic alopecia would, in theory, be halted, or maybe it wouldn't be as pronounced because there are still some other genes and enzymes that are upregulated that can also control for the expression of androgenetic alopecia. What you have to understand here is that this whole process where the genes in your hair follicles have the encoding for androgenetic alopecia can start if and only if you have the genetics for the condition, which is obvious. But I think what's not so obvious is how are these genes activated, right? Are they activated after a number of anagen cycles? Or are those genes always there in the background? And that essentially you need the right hormonal environment to trigger that kind of hair loss. And we know this from looking at studies with eunuchs. When they get their testicles removed before puberty, they don't express hair loss. And we can actually see that from Dr. Hamilton when he observed how men castrated before puberty didn't seem to lose their hair. And to be fair, this has been noticed for quite a while now, maybe thousands of years since the time of Aristotle, where he actually made that sort of observation. In any case, this makes the onset as well as the severity of the androgenetic alopecia very individualistic, as it can vary from one person to another. So, returning back to the study by Papukashvili et al., we can see that it mentions something called glycogen synthase kinase 3 beta, also known as GSK3b, which is an important enzyme Again, this enzyme is upregulated as a consequence or downstream effect of the DHT androgen receptor complex turning on certain genes that upregulate or increase the presence and activity of this enzyme GSK3B. So, when activated, this enzyme GSK3B or 3 beta attaches phosphate groups to beta-catenin, that protein that I mentioned before that is responsible for the positive hair growth effects. These phosphate groups act as a tag that signals to the cell to attach another molecule known as ubiquitin to beta-catenin. This ubiquitin tag then directs the cell's trash system known as the proteasome to break down the beta-catenin proteins. So now we have less beta-catenin signaling, which means we have less growth signals and more inhibitory signals with DKK1 suppressing the functionality of the Wnt beta-catenin pathway. So the environment now becomes more dominant for negative growth effects. You start to get more DKK1 signaling, which triggers more GSK3 beta. GSK3 beta breaks down more beta-catenin, so the beta-catenin can signal to the hair follicle cells to proliferate and grow, and this whole process is why over time your hair follicles become, more miniaturized, and it can't produce as many hairs per hair follicle, as well as the hairs themselves, their width begins to shrink and miniaturize until the point where the hair follicle can no longer produce hairs. Now, this is just fun to note, but the way that the enzyme GSK3 beta works may sound familiar because this is the mechanism of action of GT229, a proteolysis targeting chimera or PROTAC that is being developed by Kintor Pharmaceuticals with the hope of tagging ubiquitin proteins to androgen receptors in the hair follicle. Now, in theory, if this happens, the proteasomes come in and they destroy the androgen receptor proteins. And if there's no androgen receptor to attach to, that means DHT cannot inhibit those sort of damaging instructions that already exist inside of the genetics or blueprint of the hair follicle cells, right? What we have to remember is those genes are there, they just have to be triggered and activated. And if they aren't, then they remain silent. And your hair follicle, so long as it isn't too, severely damaged, can regenerate, and that positive, Wnt signaling can come in and then upregulate the growth factors that would cause the hair follicle conceivably over time to repair itself. Because now we're returning to a normal Wnt signaling pathway condition, because DHT is no longer there to kind of message, or it's not frequently there to message negative effects. So what happens here is that when those Wnt3 alpha and Wnt10 beta and all those other Wnt proteins are allowed to interact with that LRP, I think 5-6 whatever receptor, that actually leads to the downregulation or the inactivation of the GSK3B enzyme. And remember, this enzyme, when it's allowed to accumulate, it destroys beta-catenin. So if we're getting regular Wnt protein signaling and this enzyme goes down, beta-catenin can accumulate and translocate into the nucleus to promote the genes that are necessary for hair follicle development. But this sounds like a headache because, as you can see, after DHT, the downstream effects, there are so many moving parts, there are so many things to take into account. After DKK1 just attaches to the LRP56 receptor, it causes its own, downstream effects. And what you have to remember, those downstream effects are the production of proteins, right? Inhibitory proteins. And in some people, they're producing more of a specific kind of inhibitory protein, whether it's, let's say, TGF-beta or BMP, which is another protein that inhibits hair growth. You're kind of getting into the nitty-gritty here when you start to target downstream effects of DHT. So this is why we take finasteride and dutasteride to kind of, arrest the issue at the higher level rather than at the, lower level intricacies. Because it's at the lower level where you have so much variation that I don't even think you can get consistent treatment for everyone, right? You can't just target one protein and not the other, because if you target one protein, maybe your body does, some sort of compensatory upregulation of that negative or inhibitory protein, where cellular signaling notices that, oh, you have less BMP, so let's upregulate TGF beta. And all of that is mediated by someone's genetics. And it's, it's a headache. And this is where the whole individualized sort of medication approach comes into play. But at least to me, this doesn't make the Wnt beta-catenin pathway a very promising avenue for treatment, primarily because you just have too many moving parts, and each of those parts vary in significance from one person to another, such that you're not going to get a single drug or a meaningful drug that can be generalized in a population. But that's my opinion, so, take it with a grain of salt if you must. And interestingly enough, because I touched on the inhibitory protein known as TGF beta, there's actually some literature that concerns botulinum toxin, or colloquially referred to as Botox, and how it can help improve hair growth in androgenetic alopecia by suppressing TGF beta. And, there's a huge misconception. I'm not going to get into it too much, but some people seem to think that the hair growth benefits from Botox injections are due to, scalp tension, and if you can relax the scalp, then this causes for more oxygenated blood to get into specific areas. And, that's I'll just briefly address that real quick. I think people need to think this, right? If you're relaxing the scalp and there's more blood flow, wouldn't you just be delivering more DHT? Because some of the proponents of the scalp tension theory think that, because you're, having less oxygen due to high tension, which has never been proven to be definitively the case, but let's say that's the case. Because you have high scalp tension, this leads to less oxygen in the environment and more DHT is produced. Because for whatever reason, DHT is produced in low oxygenated environments, which isn't true at all. The activity of 5-alpha reductase will occur in either an anaerobic or aerobic environment, so it doesn't really matter. This whole blood flow, scalp tension, oxygen availability thing, it doesn't matter. That DHT is still going to be produced. And if DHT is still going to be produced, don't you think that if you're increasing the blood flow, you would get more free testosterone delivered to tissue sites that have 5-alpha reductase, meaning you're still going to have that DHT production? I've never seen people who kind of support the scalp tension theory address that sort of aspect. So yes, when it comes to Botox and its perceived benefits in androgenetic alopecia, it has nothing to do with scalp tension, and its benefits lies in the fact that it's inhibiting or blocking TGF beta, which has a marginal sort of benefit to the hair follicle. And we can see this demonstrated in Dr. Yuri Shan et al.'s 2020 paper titled, quote, "The Effect of Intradermal Botulinum Toxin on Androgenetic Alopecia and Its Possible Mechanism," unquote, where 18 male patients were enrolled. Those that received the injections of Botox into their scalp saw some improvements in their hair loss. Furthermore, when dermapapilla cells were observed, it was noted that the ones exposed to the Botox had lower instances of TGF beta. So right there you can see that sort of inhibitory aspect of TGF beta caused by Botox. So to conclude here on the Wnt pathway, Papukashvili et al. Shows us how blocking DKK1 protein could prevent the suppression of Wnt signaling, thus maintaining an environment that supports hair follicle growth and cycling and ensures that GSK3 beta or 3B remains inactive and beta-catenin levels remain sufficient to stimulate hair growth genes. So, what does all of this have to do with a drug that treats epilepsy, bipolar disorder, and even prevents migraine headaches? Well, sodium valproate, along with even the drug lithium carbonate, blocks and lowers the activity of GSK3 and its other variants. When it comes to mood disorders like bipolar, schizophrenia, anxiety, and even neurological conditions like epilepsy and Alzheimer's disease, GSK3 and GSK3 beta are implicated in these conditions due to their role in regulating neurotransmitters and neural activity, as explored in the review titled, quote, "GSK3 Signaling in Neural Development," unquote, by Yoon-Mi He and Feng-Quang Zhao. We need to, again, note that GSK3 beta is an important aspect in preventing beta-catenin from signaling to the hair follicle to grow, because it is the upregulation of GSK3 beta that reduces beta-catenin and allows for DKK1 to further have its inhibitory effects on hair follicle growth amplified. So, could we use topical sodium valproate to grow hair by inhibiting GSK3 beta and GSK3 activity in the hair follicle environment? Even lithium carbonate, as stated by Yoon-Mi He and Feng-Quan Zhao, is an inhibitor of the GSK3 enzyme. Well, we actually have a study that shows this being carried out to somewhat of a success. The study conducted by Seongjin Zhou and colleagues titled, quote, Topical valproic acid increases hair count in male patients with androgenetic alopecia, a randomized comparative clinical feasibility study using phototrichogram analysis, unquote. 8.3% sodium valproate in a colorless tonic spray was used by the treatment group, where the placebo used a non-active ingredient, so pretty much water and ethanol. This formulation was applied twice daily as part of the treatment regimen in this clinical trial. Now, there were 40 subjects enrolled in this clinical trial. 20 received the sodium valproate and 20 received placebo. The main outcome of the study measured the change in hair count assessed via phototrichogram analysis after 24 weeks of treatment. The study found that the sodium valproate group had a statistically significant increase in total hair count compared to the placebo group. This suggests that sodium valproate, or valproic acid, could be an effective treatment for increasing hair count in patients with androgenetic alopecia. When it comes to safety, the study reported mostly mild and self-limited adverse events, which were similar in prevalence between the sodium valproate group and the placebo group. Notably, one severe adverse event occurred in the sodium valproate group where a subject developed ventricular tachycardia. Now, this is a life-threatening heart rhythm disorder where the heart's lower chambers, the ventricles, have a very irregular beating pattern, so this could kill someone. However, the authors noted that this event did not appear to be related to the use of the sodium valproate spray, as cardiac arrhythmia is rare even when sodium valproate is administered systemically at higher concentrations typically used for seizure control. But still, the fact that this happened could mean that this person was just very sensitive to sodium valproate. But this isn't something you really want to play around with, so if you're going to do this, you have to be under a doctor's supervision, because you might not know if you are as sensitive as this person was. From a statistical standpoint, the significant difference in hair count increase alongside the comparable safety profile between the two groups supports the potential of sodium valproate as a treatment for androgenetic alopecia. However, the authors themselves note some limitations, such as the small sample size and the absence of a comparison with known effective treatments like Minoxidil. So maybe if sodium valproate went up against Minoxidil and placebo in a sort of clinical trial, you can kind of see, if it is as effective as Minoxidil or if it's not as effective as Minoxidil. Maybe it could be less effective than 2% Minoxidil, or just as effective as 2% Minoxidil, but not as effective as 5% Minoxidil. Furthermore, research with an even larger cohort and a longer duration would be beneficial to fully assess the efficacy and safety of topical sodium valproate, including potential seasonal effects on hair growth, which were speculated to impact the results in this study. Finally, I think exploring different concentrations of sodium valproate could also optimize its effectiveness, but also, it could bring in the side effects as well. But that's something that could be experimented with in the future. So overall, I think this is a solid study, a sort of maybe solid phase 1, if you want to consider that a phase 1 study. And it presents promising evidence for the use of topical sodium valproate in treating androgenetic alopecia. So perhaps maybe we don't have to use sodium valproate. Maybe we can produce a more effective protein that can inhibit the GSK3 beta enzyme. But I should also note here that sodium valproate has been documented in some cases of diffuse hair loss, so it could be the case that you are telling your hair follicles to kind of shut down too, right? But let's examine a case series titled, quote, Sodium Valproate-Induced Alopecia, a case series, unquote, by Thompson et al. The authors here report on 3 cases of alopecia induced by sodium valproate, which is commonly used for various neurological conditions. The study highlights that alopecia associated with valproate is dose-dependent and reversible as a side effect. The cases involved patients who experienced significant hair loss after long-term treatment with sodium valproate, which improved upon reduction or discontinuation of the drug. The article notes that the alopecia was non-scarring and diffuse, indicating a likely impact of the hair growth cycle without any permanent damage to the hair follicles. Each case was analyzed using the Naranjo's Causality Assessment Scale, which indicated that valproate was a probable cause for the alopecia. This was supported by the observation of symptoms of hair loss that improved substantially when the medication was either stopped or its dosage was reduced. I'm going to have a bit of a, gripe with the term alopecia being used here. It's not a hair loss per se, but it's a telogen effluvium, right? So it seems like sodium valproate, because it's not causing any sort of scarring or permanent changes to the hair follicle structure, this could just be a telogen effluvium that's induced due to the sodium valproate and its interactions in the hair follicle. I think it's also worth noting here that there may be potential side effects such as changes in mood, as this is a literal drug that interacts with your central nervous system, right? It's used to, modulate people that have bipolar disorder and other sort of, mood or depressive issues as well. So if you don't have those issues at baseline, it could kind of, mess with you and, kind of bring your mood down. Who knows, right? Liver function abnormalities and signs of increased ammonia levels has been seen in the literature when it comes to sodium valproate use, but this is debated. But it's still a concern because increased levels of ammonia can affect your brain function, and it should be monitored. And this is noted in the paper titled, quote, How to Prevent and Manage Hyperammoniemic Encephalopathies in Valproate Therapy, unquote, where sodium valproate kind of raised these ammonia levels. So overall, right, there just needs to be a better study on whether or not topical sodium valproate is 1, safe, and 2, effective when it comes to slowing down androgenetic alopecia by way of blocking GSK3 beta or 3B. So what do you guys think? Let me know in the comments, and thanks for watching this video. Now I actually have to get back to my day job because I've been spending too much time reading this script. So yeah, I'll see you guys later. Peace out.